Last speaker, Verónica Rojo. The title of the talk is Industrial Application of DNA Metabarcoding. Verónica Rojo is a project specialist at AllGenetics. She did her PhD in the evolutionary biology group at the University of A Coruña. During her PhD, she investigated the evolution of sex chromosomes and repetitive DNA elements in the genomes of laser-teeth lizards. Soon after her PhD, she joined AllGenetics, where she specialized in providing genomic-based tools to companies from the environmental, ecotoxicology, and agri-food sectors. Please.
Thank you. Hello, everyone. Well, over these 2 days, we've attended some great talks which have shown how DNA metabarcoding can be used to address multiple research questions. We've also learned a bit about the industrial applications of DNA metabarcoding. So in this last talk of the workshop, I'm going just to sum this up and outline some more examples of how DNA metabarcoding can be used and is already being used in the industry. To do so, I'm going to concentrate on 4 different fields: agriculture, environment and ecotoxicology, waste treatment and bioenergy production, and the food industry. And I'd like to start with agriculture. Well, as we've seen in the previous talks, DNA metabarcoding has revolutionized the way in which microbial communities are characterized. Of course, this has very important implications for agriculture, where we can use DNA metabarcoding to determine 2 things more easily. First, how productive crop soils can be, and second, what are the risks to plant health in a given soil. So to evaluate soil productivity, we can use DNA metabarcoding to assess the presence of plant growth promoting microorganisms, such as, for instance, fungi with activity against soil-borne pathogens. At the same time, using DNA metabarcoding can reveal the presence of detrimental species or unexpected crop pathogens, here they are, even of those which cannot be detected by culture-dependent methods. This is crucial to correctly diagnose crop diseases in order to apply the proper phytosanitary measures, which of course saves growers money. In the particular case of the wine industry, DNA metabarcoding is a tool which is more and more in demand to characterize the terroir. The terroir is the unique flavor and aroma of a wine, which depends on where the grapes were grown. So this means that wines made from the same kind of grapes, but grown in different places, have different traits. And these differential traits are of great interest for both wine producers and wine lovers. So, the terroir is not only the kind of soil, it's also the geographic location of the vineyard, the climatic conditions, and of course the microorganisms inhabiting both the soil and the grapevine itself. So in the past few years, several studies have shown that there is a strong link between the microbial communities of a vineyard and the aroma and taste of a wine. So with DNA metabarcoding, we can see how different wine-growing regions have different microbial communities. So if wine producers know the microbial profiles of their vineyards, they can better understand the unique properties of their terroirs in order to preserve the regional identity of their wines. Well, I guess you cannot see anything there, but just to give an image of how vast are the regional differences between wines growing regions. Well, after agriculture, we're going to turn to the second, actually 2 different fields of activity in which DNA metabarcoding is also becoming a very demanding tool. These are the environmental and ecotoxicology. I'm merging these two topics here because companies from both sectors share one common activity, which is characterizing and monitoring biological communities, either to assess ecosystem quality and health or to evaluate the effects of chemical agents, for instance pesticides, on the biota in water and soil. Benthic invertebrates, diatoms, soil arthropods are some of the organism groups which are commonly studied to monitor environmental change. So both environment and ecotoxicological companies encounter the same problem with these organisms, which is that they are generally identified based on morphological characters, and this is time-consuming, expensive, requires taxonomic expertise, and is also prone to error. Besides, this kind of biodiversity surveys are generally biased towards large body-sized specimens. So, DNA metabarcoding can overcome some of the limitations associated with morphological identifications. First, it may help to reduce misidentifications. Second, it makes the identification of cryptic species and immature specimens possible. And third, the direct analysis of environmental DNA through DNA metabarcoding makes the detection of a small size, rare, or elusive species easier. This means that many species which were traditionally overlooked can now be included into biodiversity inventories, therefore increasing species and the taxonomic resolution. So, in short, even though DNA metabarcoding will not completely replace the conventional biomonitoring methods, it can be implemented by environmental and ecotoxicology companies to obtain more accurate and reproducible measures of biodiversity, which in addition can be audited by third parties. Also, it makes easier the selection of novel and perhaps more sensitive indicator species of environmental pollution. And finally, of course, the possibility to multiplex hundreds of environmental samples in a single sequencing round will allow important savings in both cost and time. Well, now on to the third field where DNA metabarcoding has important applications, which is the waste treatment and the bioenergy production industry. So going back to microorganisms, the possibility to study the bacterial and archaeal populations through DNA metabarcoding has opened up a number of possibilities for the waste treatment industry. For instance, anaerobic digestion is a biological process in which organic matter is transformed in biogas rich in methane and carbon dioxide. In the last few years, anaerobic digestion has received an increased interest as a potential source of sustainable and environmental friendly energy. Currently, anaerobic digestion is being implemented in more and more places around the world to treat different types of liquid and solid wastes. So anaerobic digestion is performed by a community of bacteria and archaea that carry out different reactions to transform the organic matter into biogas. Even though the main steps of anaerobic digestion are well described, the complex microbial interactions that occur inside an anaerobic digester are still poorly understood. So, currently, the microbiome of an anaerobic digester can be compared to the black box of an aeroplane, meaning that the composition and function of this microbiome is only analyzed afterwards, that is, if things fail. So, since it is really important to understand how the microbial communities evolve during the anaerobic digestion process, biogas companies are starting to ask for DNA metabarcoding as a tool to monitor changes in the microbial communities over time in order to associate these changes to variations in the efficiency of the digestion process. So, the final goal of DNA metabarcoding will be to identify microbial indicators of optimal process performance and also to identify warning indicators of process failure. As an example, acidosis, which is one of the main failures that take place in anaerobic digesters, has been linked to shifts in the population of archaea, and actually it's been detected that the abundance of this archaeal genus increases drastically in the early stages of acidosis. And nowadays, the detection of this archaeal genus is already being used as an early warning indicator to predict and prevent acidosis. And last but not least, we have already heard this morning how DNA metabarcoding can be applied in the food industry. Well, we've all heard several scandals and cases of food fraud in the last few years, such as this species substitution in seafood and also the scandal of horse meat in beef hamburgers. Both cases had a great impact on public opinion. And indeed, according to recent studies, mislabeling in processed food products is a widespread phenomenon. And also, this poses a serious risk to consumers' health. So, the use of DNA-based methods to correctly identify the species present in foodstuffs is drawing a great deal of attention. For instance, the American Food and Drug Administration is already beginning to implement DNA metabarcoding to identify fish products. Of course, the main advantage of DNA metabarcoding in comparison with standard DNA barcoding and other conventional analytical methods is that it allows to simultaneously identify the different species present in a foodstuff which is composed of more than one ingredient. Also, as we have already learned from previous presentations, DNA metabarcoding can be applied to highly processed products, cooked, canned, powder, where proteins are usually degraded, and so protein-based analytical methods fail. So, in the food industry, DNA metabarcoding is a powerful tool to test the homogeneity of a sample, check if a species of interest is present in a mixture in order to detect a species substitution, and third, to infer the complete list of ingredients in a finished product in order to detect species that are being used as fillers and are not listed in the product label. So both Hugo and Martin have already wonderfully explained how DNA metabarcoding can be used to identify plant species in herbal products. But some other common applications of DNA metabarcoding in the food industry are, for instance, characterizing the floral composition of honey in order to know where it comes from, detect adulteration in olive oil, which is frequently mixed with low-cost oils such as soya, maize, or sunflower, verifying the species in fruit-based products such as juices. For instance, these juices labeled at 100% pure often contain cheaper fruit juices. Also to identify animal species in processed meat products such as hamburgers or meatballs. And also to verify the species of origin in dairy products such as cheese. So, as we have already discussed during this workshop, DNA metabarcoding still has some limitations and key issues that need to be improved, such as the protocols for DNA extraction from complex samples, the incompleteness of reference databases, and also the bioinformatic pipelines. But I'm pretty sure that DNA metabarcoding is becoming is going to become a routine analytical tool for quality control in the food industry in the coming years, and I'm sure that we'll see that. So, thank you very much.
I get good wine producers are asking for metabarcoding. Ribera del Duero, Rioja, are they asking for that to implement such?
Yeah. Also, there are already some ongoing research projects, and of course, wineries are also involved because, the influence of the microbiome, both in the cellars, but of course in the vineyards and the grapevine itself, has a big influence on the final organoleptic properties of wine. So yeah, wineries are already in Spain are starting to be involved.
Increasing.
Yeah.
Okay.
So regarding using metabarcoding in food production and quality control, is it being used to check for pathogenic organisms in food? I'm thinking of those cucumbers, German cucumbers that caused an outbreak of some severe infection some years ago.
Yeah.
I wonder if metabarcoding is becoming more and more common in this quality control pipelines in food production in order to check for pathogens, not just for the...
Yeah, exactly. I didn't include this topic here to stick to the time for the presentation. But DNA metabarcoding is already being used to monitor periodically both the surface where the foodstuffs are prepared, and also the final products, and also to compare between the efficiency of different treatments to increase shelf life. So of course, studying the microbiome in both raw materials and the finished products is already being done through DNA metabarcoding. Yeah.
I'm curious about, because that's what we see sometimes with these plant products, that the main product is there, but also if we take two different spices that are, say, labeled as oregano, but then there is a lot of background also, like other plants present, but sometimes they're very specific to particular geographic origins, so you can actually tell that this was produced in Asia, for instance, and this one produced somewhere else. And I'm wondering about the wine industry, because I guess that it's quite often also a case of like adulteration, that you claim that it's Tempranillo and then you add some other variety or so. Is it any work that you are aware of, like to actually narrow down the maybe not geographical location, but this particular strains of it?
Not really. I mean, I'm aware of studies to verify the grapevine variety. But exactly the geographic origin in wines made from the same variety, I'm not aware of that kind of studies, but it would be a good line of research there.
It is a completely off-topic question, but if we are being cheated by the food companies about what we eat, and you may know like how general this is, like if they are cheating us a lot or only a little bit, how much expensive things are going to be when we will not tolerate that cheating?
I'd like to have the answer for that.
More or less, like if we are being cheated a lot, like prices will go very high. If we are just a little bit like.
Well, maybe not necessarily. I'm not sure, but I think that the regulation and will probably become more strict when these kind of technologies are implemented. But well, yeah, maybe it's an additional analytical step for food companies, but I'm not sure if this, because I mean, there is a margin. I'm not sure if this will translate directly into a price increase in the final to the staff. Hope not.
Well, I don't know, is that maybe people will not be very happy with.
Yeah, no, but for instance, if you're paying, I mean, a fortune for a bottle of olive oil, you want olive oil.
Yes,
so this profit that companies are having is because they are lower cost species or some oils. Well, they will reduce the profit margin, of course, but I don't think that the price of the final product can go any higher. So, I mean, just having what you pay. I don't know, but I have no idea.
Like this is going to be a revolution. Like the food industry will be showing us what they are selling us, and they will have to change what they do. Like our shopping bag will change, and the money we pay for those things, I guess.
Finally, the market controls that. Right? Sorry, I cannot hear you properly.
How often is DNA metabarcoding is being demanded by private laboratories to do clinical diagnosis?
Well, based on our experience, nowadays it's more that I don't know in the herbal industry. Probably Martin has a better picture of that, but it's still in a very preliminary stage.
So main hospitals, public hospitals, they are not using the
you're talking about the, sorry, I think I didn't understand your question.
health science and clinical diagnosis.
No, yeah, for clinical diagnosis, and probably Marga and Juan can answer also this question.
It's already being demanded by private companies as a tool for clinical diagnosis.
Well, I'm not expert in the clinical sector. I know in the public hospital, for instance, and other research groups, yeah, it's already very demanded. And in the private sector, I'm not very aware of the clinical companies, but at least in the food industry, yeah, it is starting to be demanded because it can solve, it can answer questions which previously couldn't be answered.
But I guess in this case it's a question of time, because sometimes in hospitals, I guess they need to have the answer very soon, so it would be better to use qPCR, but perhaps to study the whole diversity and then do qPCR tests.
Yeah, exactly. This is frequently the common approach. I mean, you first want to know what you have in sample, the whole composition, to select indicator species which you can detect using a quicker approach as qPCR, as Joaquin said, which is more or less the same what happened in the anaerobic digester. First, you want to have a complete view of the whole composition of your microbiome, and then to find a proper indicator that you can detect in a quicker and cheaper way, because of course the timeline for DNA metabarcoding projects is several weeks at least, and sometimes this is too long for a quick diagnostic.
Yes, and let me answer just to support. In hospital, the time is key for everything, for live patient, and you cannot rely on such technology that has a huge potential for industrial applications. but not so far for rapid clinical diagnosis, so far. Another way will be the outbreak tracking or outbreak follow-up in outbreak hospitals to see the one strain is the same as the other. So that is another issue. But for diagnosis, in 1 hour you need to administer antibiotic or not. So far, this technology is not for this purpose. But in the future, we will see if we can rely on this rapidity. Okay.
Of course. I mean, sometimes you need to have the results in a 1 day time lapse, so you cannot wait for the results of DNA metabarcoding. But for exploratory purposes, to have the complete view of a catalog of a species present in a sample or in a given moment, it is useful for that. Okay. Well, thank you then.
That was our last talk of the workshop, and I hope you've enjoyed it. I hope you enjoyed our social activities as well. I hope you've had time to do some networking, and hopefully some ideas have come out of there. And I just wanted to thank you all for coming, especially the speakers for some awesome talks and some pretty fruitful discussions. I would also like to thank the people from the INIBIC and from the group from the evolutionary biology group, and my colleagues from AllGenetics for the organization. And yeah, thank you all for coming.